Thank you very much for this really encouraging data. Are there any urgent questions at the moment? Otherwise, we have the chance to discuss the talk afterwards in the panel. So thank you very much again.
Thanks.
And I would like to announce now Cristianne Rijcken. Cristianne Rijcken is the CEO of the Cristal Therapeutics pharmaceutical company. Cristal Therapeutics company is a member of ETPN. It's very important because I would like to encourage also stakeholders here being a member of ETPN because this is essential for demonstrating nanomedicine and the efforts of nanomedicine for the benefit of the patient, especially in front of the European Commission, because if we don't convince the industry being really interested in nanomedicine, we won't be successful at the end. And so therefore, I'm looking very much forward for the talk from Dr. Rijcken.
Thank you very much for the kind introduction. Indeed, it's a pleasure to present Cristal Therapeutics to you, and I thank the organizer for letting me do so. So Cristal Therapeutics is developing CRIPEC-based nanomedicine, and what we are is that we are a nanomedicine company, a small company as to say. But we are originally founded in Utrecht, a spin-off of the Utrecht University, but since half a year approximately we've moved to Maastricht, which is the south of the Netherlands. The company is funded by venture capital, Dutch VC as well as some regional VCs, and as such the company is managed by Joost Holthuis as well as myself. So that is some of the background, so the company background, but of course it goes about the products that we are developing. And as indicated, CRIPEC is our proprietary platform, Cristal Therapeutics Polymer Technologies, and based on that we can develop multiple products in different disease areas, so meaning in oncology as well as also in chronic inflammatory diseases. But our first product is CRIPEC docetaxel, so the combination of our platform, an nanomedicine platform with docetaxel, and we aim to go towards a phase 1 study early next year. Next to our own product development, we're also developing with other parties products together. So then to immediately dive into the topic of nanomedicines, we have seen different type of nanoparticles being generated, but I'd first like to show you how we make our nanoparticles, and I think we make them in a fairly unique manner, namely a two-step process to create what we call transiently stable nanomedicines. So that means that we take a drug molecule, we derivatize it with a linker molecule, so you create a chemically derived molecule. And if that is then combined with a proprietary polymer, you initially get micellar structures, but we subsequently kind of stabilize that structure by making these so-called cross-links, so really bridges between both the polymer as well as the drug molecule. And as such, this is actually one big macromolecule, as everything is covalently linked to one another. However, in time, the linkage between the drug linker as well as the bondage between the polymers, they are being cleaved. And then in time, you have the native drug being released again. So really the native drug molecule, so there's no linker fragment being attached to it, and that can then exert its activity, while at the same time as indicated the polymer composition as such also disintegrated into smaller fragments, which will ultimately be cleared by the kidneys. And I think this approach is different versus, for example, liposomes, which is a reversible system, as well as traditional nanoparticulate systems. As by this approach, we can really tune the release rate of the drug. So other unique features are really the tunability of the system in the sense that the biodegradability element can be tuned. So by using different type of linkage between the polymers and the drug molecule, we can have systems that degrade within a few days up to several weeks. The tunability is also corresponding to the size, so we make very monodispersed particles, but we can make them at least as small as 30 nanometers up to at least 100. We've successfully used this approach for a whole range of molecules, so small molecules, peptides up to 40 amino acids, so fairly large. Hydrophilic, hydrophobic has all been successfully used. And moreover, we've also really can tune the release profiles, and then you can consider, and I think it was one of the previous talk by Selecta, by using different type of compounds within the nanoparticles, but then each with their own release profile, so really having the best exposure to the drug in time. We can modify the interior part, and we can also modify the exterior part by using chemistry That allows for ultimately also active targeting. And not unimportant is also the ease of manufacturing, which I'll show you in the next slide, as normally liposome formulation is rather complicated. I heard recently that Doxil takes more than 17 steps to actually make the product, while in this case it's a really two-step synthetic procedure. So first you make your drug linker, chemical step, and then you make your nanoparticulate structure, which is a chemical step as well, followed then by the ultimate aseptic filtration to get your drug product. And this is also to show you a picture of how the cryoptic dose Taxol looks like, indicating also the nice monodispersity of this approach. We have already successfully upscaled towards GMP production, and also the process is very scalable. And also, as indicated by the subtitle, the production process of these nanoparticles independent of the type of API. So, irrespective whether it's a small molecule or a peptide, it's the same process that can be used. The platform as such, well, it's nice to have a technology, but at the end of the day it's about making products, and that's what we're making the nanomedicine for, is to really improve the therapeutic profile by having a stepwise approach, having first assurance of a prolonged circulation in the bloodstream. That will thereby we aim to improve the disposition in the body, meaning not going to the healthy tissue but really focusing to go to the target tissue, and even further, if you could go for active targeting, you could have an increased cellular targeting. And of course, if the nanoparticles do reach the tumor site or even intracellularly, you need to have a tunable release in time. So what we're developing ourselves is cryoptic dose Taxol as being the first product, as indicated. We're developing it towards gastric cancer with the aim to go to phase 1 early next year. Dexamethasone is a compound that we have proof of concept with. We haven't chosen the exact indication, but we'll also take that forward. And next to that, we also co-development with other parties the platform towards the active targeting approach, as well as we're combining a peptide from Nexogen into our nanoparticles and take that forward. So then to show you some data of cryoptic dose Taxol. What we have done is in the breast cancer studies with, and I'd like to point your attention to that, studies with only one single injection. So normally you see these kind of efficacy studies where you have multiple injections before you see a therapeutic response. What we've done here is at the equimolar dosage we administer Taxotere, so the current product, and the cryoptic dose Taxol, so to have a really equimolar comparison. And as you see, if the tumors are fairly small, then you give the drug. Taxotere does result in a regression, but then also it relapses fairly fast again. While in case when we give cryoptic dose Taxol, you see a really long-term suppression of the tumor growth in time after, as indicated, only one single injection. Because of the improved safety data, and I'll show you some of those safety data later on, we could give a higher dosage, and that's depicted in this plot where we dose Taxotere 30 and cryoptic dose Taxol 90 milligram per kilo. And then we let the tumors grow until they're fairly established. But then even after only one single injection, you have a complete tumor regression. So I think this is a fairly major improvement versus Taxotere. And if you would ask me what is the reason for this, is then I would depict you to these figures, namely the tumor accumulation in time. So if you dose equimolarly, so to allow for a fair comparison in time, we measured after 2 or 4 days the accumulation of dose Taxol in the tumor site, and as you see, there is approximately 24 higher amount of dose Taxol that have reached the tumor in case of cryoptic dose Taxol versus Taxotere. When you could even give higher amounts, so more of the cryoptic dose Taxol, then this differentiation is even bigger, as illustrated here, that even after 7 days you see an enormous amount of cryoptic dose Taxol that have reached the tumor site. This is from the efficacy perspective, the studies that have been performed in mice. What we've done also is the part of the preclinical safety package, the PK studies, and they nicely illustrate the PK profile versus Taxotere. So what was done here, administration to healthy rats, Taxotere giving the normal pattern of fairly rapid elimination, while in case of cryoptic dose Taxol, where we dose slightly higher because of the improved safety profile, we measured in total dose Taxol, so that's the representative of intact nanoparticles. And as you see, they're really long circulating through the bloodstream, having a half-life of approximately 15 to 17 hours. This was part of the preclinical safety program, but additional data were gathered in acute as well as 5-day tox studies. And for those who aren't familiar with that, with a 5-day tox study, what you do is you give every single day for up to 5 days an injection of the compound to get an idea which organs are affected most on the long run. What we saw out of these data is that cryopec empty, and that means literally that there was no drug involved, but you have the exact same nanoparticles, that there were no biological variations versus vehicle. In other words, the particles are fully safe. The MTD of cryopec dosed XL was significantly higher than Taxotere. And what we also measured in the 5-day tox study was that we do observe side effects, but those were of significantly less intensity. And I've elaborated on that in this slide even further, where I like to point your attention to the dose that is given. So Taxotere is giving at its MTD in rats, while we gave approximately 140% higher dosage because, as indicated, of the higher safety. But then still what we measured in time is that we have, for example, the most dose-limiting toxicity, myelosuppression, significantly less effect on the white blood cells. So that's, of course, all preclinical data, but for us it's very convincing and promising to go forward towards a phase 1 study. And actually we're now doing the subchronic study, so that is dosing at a clinical, more logical regimen every 3 weeks, dosing and monitoring the rats, how they behave and where any potential toxicity is being observed. That being the step before going towards the phase 1 study, which we scheduled as a fairly straightforward phase 1 safety evaluation, then followed by phase 2A, where we will go and monitor the efficacy in a gastric population, but also potentially another indication depending what comes out of the phase 1 study. And for this, we have already received positive feedback from the European authorities. So having said that, I also like to make a comparison what we are doing versus the other nanoparticles, nanomedicines that are being developed, and we've also heard some lectures earlier today and yesterday. I think what we have developed is a platform that allows for really generating superior efficacy in different preclinical models. I didn't show you the results of the dexamethasone, but there we also saw upon equimolar administration really superior activity because of the really enhanced accumulation at the target site, while at the same time having a completely superior tolerability profile. Because of the ease of manufacturing, it also allows for very fast new product development. So really the design and the production of a new product is very straightforward. Having said that, it's a brief snapshot of what Cristal Therapeutics is doing, and as indicated, we hope to go towards phase 1 early next year, so maybe next year I can already give you a short update on that. And I'll be more than pleased to talk about any collaboration later on. Thank you.